Hello everyone, and welcome back to AP Bio with AP Playground. Today we're covering topic 5.1, meiosis. As the title suggests, today's topic is all about meiosis, and we'll be looking at all of the steps that make up meiosis, including both in meiosis 1 and meiosis 2, and we'll end by comparing mitosis and meiosis. And our suggested skill is Concept Explanation 1B, explain biological concepts and processes. So, sort of background on genetic transfer. Genes are segments of DNA that code for heredity and are transferred generationally through reproduction from parent to children. And reproductive cells that transmit genes are known as gametes, and these are opposed to somatic or body cells. And the gametes that humans have are going to be sperm and ova. And asexual reproduction is going to be when a single parent passes their whole genome to the offspring. And this is going to happen through mitosis, where we're generating an exact clone of the cell, versus sexual reproduction, where two parents contribute genes to the offspring, leading to genetic variation. And this is what's going to happen in meiosis. So instead of creating an exact duplicate of the cell, we're now changing up the genome of the cell through genetic variation. And it's important to remember that homologous chromosomes are going to be similar chromosomes, and we get one from each parent. And we can see this in a karyotype, which is right up here. And we looked at this diagram when we were discussing topic 4.6, meiosis. Now, there are going to be 23 pairs here for humans, which make up 46 chromosomes, and human somatic cells, which are non-sex cells or body cells, these are all going to have 46 chromosomes. And they're going to be a diploid cell. And then cells that emerge from mitosis are also going to have 46 chromosomes because we're creating a direct copy of the cell. And sex cells, gametes, are going to have 23 chromosomes. They have half of the genetic information that's known as a haploid cell. So diploid cell can be represented by 2N, while a haploid cell is just N. And it's important to remember that from these 23 pairs, 22 of them are going to be autosomes, which are non-sex determining chromosomes, while pair 23 is going to be a sex-linked chromosome, such as an XY or XX chromosome, that is going to determine the person's sex when they're born. And during meiosis, one chromosome of each pair is given to a gamete randomly. So each gamete cell is going to have half of the genome. And remember, that's the whole point of meiosis. We're trying to create cells that when they come together will form one full genome. So that's why each meiosis cell is going to have 23 chromosomes. And then fertilization is when egg and sperm are going to come together to form a fertilized diploid zygote. So essentially, when we take our meiosis cells and that are all going to be haploid and have 23 chromosomes each, and we put them together to form our diploid zygote, which is going to have 46. So you can see up here, we're in the diploid phase. We have diploid cells, we're diploid adults, then we go through meiosis, and now we've got our two sex cells, our two gametes, that are then going to be fertilized to go back to a diploid zygote. Now, like I mentioned in the objectives, meiosis takes place in two series of steps, and the first one, very intuitively named, is meiosis 1. And meiosis 1 technically begins during interphase, where during S phase, our DNA is going to be replicated, just like in mitosis. But unlike mitosis, we have two stages of cell division. So we have 4 daughter cells instead of just 2. And meiosis 1 begins with a diploid cell that has 2N equals 46, where we have 46 chromosomes. And remember that during this synthesis phase, we stay with 46 chromosomes. We have let's say we have our first pair of homologous chromosomes. One came from the father, one came from the mother, and each of these chromosomes is going to gain a sister chromatid. So for one pair of homologous chromosomes, we now have 4 sister chromatids. So we still have 46 chromosomes. We just have the amount of chromatids has been doubled. And so we're still beginning a diploid cell. but now each of these chromosomes has been replicated with a chromatid. And during prophase 1, which is part of meiosis 1, the chromosomes condense so we can distinctly tell each chromosome, and we have what's known as crossing over, where these homologous chromosomes, each with two sister chromatids, are going to be attached in synapsis, and then there's going to happen crossing over, which is when DNA from one homolog chromosome is cut in exchange with the exact portion from the other one. And that's going to form crisscross regions known as chiasmata. So here at crossing over, this portion of each homologous chromosome is going to be cut off and swapped. And here, now we see genetic swapping. Now, this is the father's chromosome with a piece of the mother's genetic information, and vice versa on the other homologous chromosome. And you can see here in our prophase 1 that we have developed these chiasmata, which are these points where crossing over has happened. And at metaphase 1, all of the homologous chromosome pairs line up at the metaphase plate, just like in mitosis. However, the important thing is that there is independent assortment. They can line up in the sense along this metaphase plate where the majority of the dad's genetic information is on one side of the metaphase plate and the mom's is on the other, or vice versa. So there's sort of a 50-50% chance here that one side of the cell is going to get mostly mom's genetic information plus whatever crossing over happened, and the other one will get mostly dad's genetic information plus whatever crossing over happened, and that's going to help increase genetic variation. Now, in anaphase 1, the homologous pairs separate and move toward the poles, but the sister chromatids stay connected. And this is different from mitosis, where sister chromatids separate during anaphase. So we're only separating homologous chromosomes, but sister chromatids are still staying together. And during telophase 1, we reform the nuclei and cytokinesis occurs. So now, at the end of meiosis 1, we've reached two daughter cells that are all haploid cells. They only have 23 chromosomes each, but each one has a sister chromatid as well. So now begins meiosis 2, where we start with our haploid cell, 23 chromosomes, but there are also going to be sister chromatids. And during prophase 2, these sister chromatids are going to move toward the metaphase plate. Then metaphase 2, they're arranged on metaphase plate as per usual. And of course, these sister chromatids are not going to be all the same, because you see here, there's been crossing over that's happened. So they're not, each sister chromatid isn't going to be identical to its other sister chromatid. And in anaphase 2, just like usual in anaphase, these sister chromatids are pulled apart and moved to opposite sides of the cell, and then during telophase 2, the nuclei reform, we have cytokinesis, and we end up with 4 daughter haploid cells, all genetically different because of the crossing over that has happened. So lastly, it's important to make a list of the differences between meiosis and mitosis. Obviously, in meiosis, there's going to be synapsis and crossing over, versus mitosis, where there is no genetic swapping. There's no forming of the tetrad, which is where the 4 sister chromatids come together at the chiasmata points. There's not going to be any of that genetic swapping because we're trying to create genetically identical cells. At the metaphase plate, the homologous pairs are positioned and then they're not individually replicated chromosomes like in mitosis. We're not pulling apart just sister chromatids, we're pulling apart homologous chromosomes that have formed in these tetrads. And lastly, sister chromatids stay attached during the first round of anaphase in meiosis, whereas in mitosis, there's only one round and these sister chromatids are being pulled apart during it. So here, mitosis is going to have 4 stages. PMAT plus interphase is going to happen in somatic or body cells. The purpose is just to create more cells for growth, for tissue repair, for asexual reproduction, and we're getting 2 diploid daughter cells. Chromosome number is going to remain the same, and genetic variation doesn't change. Whereas the meiosis has 8 stages in total. We're going through 2 rounds. It's happening in germ cells. These are our gametes. The purpose is sexual reproduction. We want genetic variation. We're getting 4 haploid cells. The chromosome number is halved in each daughter cell. We go from 2n to n. And lastly, genetic variation has been increased through crossing over. And They are same in some cases because they are producing new cells. They're similar basic steps and they start with a single parent cell. But overall, meiosis is the one that's going to be causing our haploid cells that have genetic variation, while mitosis is causing diploid, genetically identical cells. So there you have it. Meiosis versus mitosis and the steps of meiosis. We've got that crossing over happening causing genetic variation, and we go from one diploid cell to 4 of these haploid cells. And meiosis versus mitosis, no genetic variation in mitosis, and we go from a diploid cell to a diploid cell. And there you go. That's 5.1. See you next time.